every spring, as temperatures gradually rise and life springs to life. A stunning pink and white blossom quietly blooms in urban parks and mountains across East Asia and across the globe. This blossom is the widely known cherry blossom. Cherry blossom is not a specific species, but rather a collective term for a variety of ornamental species and their horticultural varieties within the genus Prunus in the Rosaceae family. Famous worldwide for its brief yet dazzling bloom, it not only embodies a profound cultural heritage, but also holds a wealth of scientific knowledge about its biological characteristics and ecological adaptations. From a plant taxonomic perspective, cherry blossoms primarily originated in East Asian countries such as China, Japan, and South Korea. According to the flora of China, there are over 30 wild cherry blossom species native to China, including mountain cherry, winter cherry, and bellflower cherry. These wild species are the genetic source of many current ornamental cherry varieties. After years of artificial breeding, particularly the systematic cultivation of cherry blossoms in Japan since the Edo period, there are now over 300 registered cherry blossom varieties worldwide. The Sumi-yoshino is the most famous and is widely planted in urban greenbelts. The cherry blossom blooms for a very short time, typically lasting 5 to 7 days, with some varieties blooming for only 3 to 4 days. This phenomenon has clear biological reasons. Cherry blossoms are typical flowers before leaves plants, meaning the flowers bloom before the leaves unfold. This strategy helps improve pollination efficiency. Without leaves obstructing the flowers are more easily visible to pollinators, and sunlight reaches the flowers directly, raising the local temperature and promoting antidehiscence and pollen dispersal. However, the flower structure is very delicate, with thin cell walls in the petals, which easily lose water. Exposure to wind and rain or drastic temperature fluctuations can cause the petals to shed rapidly. Scientific research has shown that a specialized layer of cells called abscission layer exists at the base of cherry blossom petals. When pollination is complete or when faced with increased environmental stress, these cells secrete enzymes to break down the cell walls, causing the petals to shed. While this mechanism shortens the flowering period, it effectively avoids energy loss and focuses resources on fruit and seed development. Cherry blossoms have extremely demanding climatic requirements, and their blooming time is closely linked to temperature fluctuations. In meteorological research, accumulated temperature models are a key tool for predicting the first blooming period of cherry blossoms. Specifically, when the daily average temperature is consistently above a certain threshold, typically 5 degrees Celsius, the plant begins to accumulate heat. Once the effective accumulated temperature reaches a certain value, the flower buds begin to germinate and eventually bloom. For example, the Sumi-yoshino cherry blossom typically requires an effective accumulated temperature of approximately 600 degrees Celsius per day to bloom. For this reason, cherry blossoms are considered a key phenological indicator plant, and changes in their flowering time can directly reflect the direction of climate change. Since 1953, the Japan Meteorological Agency has continuously recorded the blooming date of cherry blossoms in Kyoto, Tokyo, and other areas. Data shows that over the past 70 years, the first blooming period of cherry blossoms in Tokyo has advanced by an average of approximately 0.37 days per decade. This trend is highly consistent with observations of rising spring temperatures in the context of global warming. Cherry blossoms also occupy an indispensable position in the ecosystem. Although most ornamental cherry blossoms have low fruiting rates due to long-term artificial breeding, wild cherry blossoms are extremely important nectar plants. Their flowers contain rich nectar and pollen, attracting numerous pollinating insects such as bees, bumblebees, and hoverflies. Research results showed that the sugar concentration of cherry blossom nectar generally ranges from 30% to 50%, mainly composed of sucrose, glucose, and fructose, providing a vital energy source for insects active in early spring. Furthermore, cherry blossom leaves served as food for the larvae of certain lepidopteran insects, playing a role in the local food web. It is important to note that the ornamental value of cherry blossoms is closely linked to their aesthetic color. The color of cherry blossoms is primarily determined by the pigments in their petals. Pink and red are due to anthocyanins, while white is due to the lack of pigment in the petals, revealing the natural color of the cytosolic sap. Anthocyanin synthesis is affected by light, temperature, and pH, so the color of the same cherry blossom variety may vary slightly under different environmental conditions. For example, The flowers of the bellflower cherry blossoms become more vibrant and eye-catching in warm, and humid environments. Modern molecular biology research that has identified several key genes that regulate cherry blossom color, shape, and flowering time, laying a solid theoretical foundation for genetic improvement. In China, the cultural and scientific value of cherry blossoms is gradually gaining increasing recognition.